Okay, I think it's time that we can get started, everyone. I'm glad to see several people have said hello in the chat. If you've never used Zoom before, you'll see that if you look at the screen you're looking at right now and you just touch it with cursor, you'll see at the bottom there is a menu bar. And in the menu bar, you can see there's a place that has a little cartoon bubble where they're chat. And what we're doing right now, Cassie Sofing, who works with me on Globe Mission Mosquito, we're both online and doing the presentation. She'll be answering questions in the chat. So feel free to, as things come to your mind, go ahead and ask us about it. And then when she's online, I'll do the same thing. So these two of us sort of trying to interact with you at the same time. So let me introduce myself. My name's Rusty Lowe. I'm scientist lead for Globe Observer Mosquito Habitat Mapper. I live in Boulder, Colorado. I work with the Mission Mosquito, which is run out of Goddard Space Flight Center. And Cassie, would you like to introduce yourself, please, as well?
Hi, I'm Cassie Sofing. I'm actually based out of Sioux Falls, South Dakota, and I'm a former middle school and college teacher, and remote sensing is my interest area.
We have a lot to cover today, and we're not going to be able to cover everything, but the idea today was just to have you put some faces with names and get going, and we have several ways that we're going to communicate, and we'll talk about that in a minute. But now I'm going to show you some slides that's going to help introduce you to our project today, and let's see what you think. So here we are, and I want to welcome you to Globe Mosquito Habitat Mapper and the virtual research cohort this summer for Globe Mission Mosquito. And I want to welcome you. I want to tell you how excited that our whole team is that you have decided to join us in this activity. I think it's going to be very exciting. We're doing research that no one's done before, and that, I think, is what makes science so exciting. So what we're going to do today is basically just three things. We're going to talk a little bit about why we're doing what we're doing, present some of the activities that we hope to be doing over the next 2.5 months. Together, we'll build a larva trap like that you will want to use around your house. And then we're going to have a question and answer. So I hope that we can cover all that in 45 minutes or so. I don't want to keep you too long. It's for some people it's lunch. Some people have just gotten up. Thank you so much for being here. So what I wanted to do is just start by talking about why we're in. And this is a tapestry that was created by one of the artists that I work with. And it really describes, I think, in a very textural way, why mosquitoes are such an interesting thing to study, because literally every kind of is I'm not sharing. So anyway, what I did is I basically went over the outline of what we're going to be talking about. We'll talk about the problem. We're going to talk about what we're going to do together. We're going to build a larva trap. We'll have questions and answers. Okay? So apologies, great apologies for you. We're traveling today. We're doing several face-to-face workshops, so we're not in our own space, so I'm a little discombobulated. Okay. Anyway, I wanted to tell a story about mosquitoes and why I'm so interested in them. And the story is really the mosquito story is a climate story. And I don't know if you have learned much about the people on the team yet. I'm learning just a little bit about you and your posts. But I started out in the field of paleoclimate, which means that my interest is understanding what climate was like long ago. And that's really important, because if we didn't have those long records of climate change, we wouldn't be able to know what we're seeing today and why it's important. What we're seeing today in climate change is the rate of change is now far faster than we have seen in the in the past archives of change that we look at, the lakes and the ice cores and the ocean cores. These are the places where we can tell what the world used to be like. And the rate is faster, and the amount of CO2 that we have in the atmosphere is higher than it's been any time since the Pliocene, which is more than 2 million years ago. And this is why we are so concerned about climate change. And but everything is connected to everything else. And one of the things that happens in climate change is organisms are also responding to changes. And that's what we're seeing now today with mosquitoes, because they are now moving from where they used to be to new habitats, as new habitats are having changes in the environmental conditions so that they're now able to support these new species. And that's why we have invasive mosquitoes in the US right now. But I wanted to go back and tell a longer story. And the story is about why we have these mosquitoes and how they get around. And what's very interesting to me is that for this old problem, we can apply very new techniques, like the techniques of looking at gene sequencing. And people have worked on the mosquito genome, and we know that the mosquitoes that we have in the United States today are mosquitoes that have come from South America. But we had also the same mosquito, the Aedes aegypti mosquito, which is one of the really serious mosquitoes. It is the one that's responsible for diseases like dengue, chikungunya, yellow fever, and of course Zika. And one of the reasons we started this project was to monitor these mosquitoes, as well as mosquitoes for things like West Nile virus that are problems today. But anyway, the big story is that these mosquitoes have changed evolutionarily over time. You'll see over here in the lower right, a tree. Well, these mosquitoes used to like to live in tree holes. They're called tree hole mosquitoes, and they live in containers. And what happened was that they were talking about 10,000 years ago. That's a long time ago. But in 10,000 years ago, the Sahara Desert wasn't as expanded as today. And we actually had mosquitoes running all the way across the northern part of the Africa continent. And then through these last 10,000 years, we called them the Holocene. What happened was it got wetter and then it got drier. And so after about 7,000 years ago, it began to get drier and drier and drier. And what happened is these mosquitoes that used to live in these tree holes began to come more and more and more in contact. And people which were also distributed across the landscape, were also beginning to come together and live in river valleys and places where there were water. So because of changes in climate more than 7,000 years ago, people and mosquitoes were brought together in the same place. And these mosquitoes discovered, hey, why would I put my eggs in a tree hole when all around these human habitation sites, there are containers? People are making these, baskets, and lining them with pitch, and they put water in them. And this is a great place for me to put my babies. Also, pottery, all these other different kinds of containers that were there. And so over time, the Aedes aegypti mosquito, which we also call the yellow fever mosquito, adapted, and they became what we call anthrophilic. They love to be around people, and they bite humans preferentially, and they lay their eggs preferentially in the containers that we provide. Today, those containers are not, beautiful clay pots, but they might be discarded McDonald's cups. Anything that can hold water outside is where they're going to put their mosquitoes, and anything that holds water inside is where the mosquitoes are going to put their babies. So these mosquitoes then made it to the New World through the explorers and the colonists and the slave trade. And these mosquitoes were in the containers of water that were brought overseas. So that was the first time that we got the Aedes aegypti mosquito in the New World. We eradicated those mosquitoes using DDT in the 50s, and we thought we were done with them. But now, as climate is now changing, they're now moving northward, and we're now seeing them once again reestablishing in the North American continent. And last but not least, another way that these mosquitoes are traveling is up here on the left, is because we have so much travel. People are moving around, and people are moving around and bringing mosquitoes with them. In the case of Zika, they could be carrying the disease that mosquitoes carry in their own bodies and transmitting it, and that's really serious. The other mosquito we'll be talking about today is the Asian tiger mosquito. And the Asian tiger mosquito wasn't in the New World, north in the Americas, until the 70s. And they got there because they found the containers that humans create also to be marvelous nurseries for their young. In this case, it was tires, the used tires that were going across the ocean and being sold. And so in the 1970s, we began to get these two mosquitoes, and now we're finding them all over. So here, if you think the Asian tiger mosquito used to only be in Southeast Asia, and now you'll see that is the what we see the yellow here in this map. And the red is the mosquito that used to only be in Africa, the Aedes aegypti, the yellow fever mosquito. You can see it all over in the red of the map. And where you see orange is where those mosquitoes both can be found. And without giving you too many graphs and too much detail, what you need to know is that both these mosquitoes are now moving northward. And they've been moving northward by a few kilometers every 10 years or so. And so by monitoring the mosquito habitats that you have where you are, we can begin to understand where those mosquitoes are going, and we can put it in this long time scale of what these mosquitoes were doing since for the past 10,000 years up to the present, and why, and we can prepare ourselves for the kinds of issues that might come because they can carry disease today. So that is essentially the mosquito story that caused us to think about creating the mosquito habitat mapper in our lab. So I don't know how many of you have a strong earth science background. Earth science is my favorite science, but I know when I was in high school, I took chemistry, I took physics. You know, I didn't get to take earth science until 12th grade as an elective because it wasn't required. But really, I think it's one of the most exciting sciences because you have to know chemistry and physics to understand it. But the most important thing about the Earth system is that everything is connected to everything else. And so if you change something in the atmosphere, like carbon dioxide, it's going to change life. It's going to change water. It's going to change even the lithosphere, which is our rocks and our soil. And so by monitoring the atmosphere, the rocks, water, and life, we can begin to better understand how changes in climate are propagating and changing all different parts of the Earth around us. And so if you look at the Globe Observer app that you see here on the right-hand side, you'll see that we have clouds, which is the atmosphere. You'll see that we have land cover, which is this biosphere. Mosquito habitat mapper is, of course, life, so it's biosphere, but it's taking place in water, so it's hydrosphere. And so we are basically trying to interlink and understand how these different parameters that we can measure using Globe Observer, how they're interacting with each other. So this is a little bit of a spoiler, but when you do mosquito habitat measurements, we're going to ask you also to do land cover measurements, because that will help us understand, why are the mosquitoes there? What is the environment like? What is the precipitation like? What is the temperature like? You know, what kind of things are living there? What's the soil like that causes those plants to be there? So we're going to be able to have a nice context, an environmental context for your mosquitoes just by doing the mosquito habitat mapper measurements alongside land cover. And we'd love you to do clouds as well if you're interested. All of the apps work a little bit the same way. And if you can do one, you can basically understand how to do all of them. And we'll talk a little bit more about that. If anyone has any questions so far, please put them in the chat, and we'll try and take a look at those as we move forward. I've covered a lot of information already, and I haven't even talked about what we're going to do yet. But I thought it was important to just put a little bit of a context so that you know why we think this is so important from a science perspective. Of course, there's also the health perspective that we'll talk about in a minute. So Cassie just asked how many of you have downloaded the app, and so we'd love to know how many you have. Cassie, you were just looking at the scientific visualization. How many measurements, or how many folks do we have so far that have actually downloaded the app and made a measurement yet?
We have 27 that have joined the SEES team, and they've made a total of 11 observations, and most of them have been clouds, so that's a good way to get started, and it was
Absolutely.
one with mosquito so far.
Perfect. So thank you so much for, we were hoping that at least some of you would have some experience with the app, not because you need it for today, but because then if you've had some experience, you know what questions to ask while you have us, here front and center. So do let us know as you move forward when you have some issues. But right now I want to tell you why we're doing Globe Mission Mosquito. And the problem is that mosquito-borne disease is huge. It causes more human deaths than any other organism in the world. And so more than almost 1 million people every year are killed by mosquito-borne disease. Many, many more are impacted by the disease. And so as a result, it's something that we need to pay a lot more attention to. And up until now in the United States, since the 1950s, we haven't had these deadly diseases in the States with any strong frequency. But now, because of the change in climate, because mosquitoes are adapting, because new diseases have come, we're now getting very serious mosquito-borne diseases in the US as well. So using the power of citizen science and using the Globe Mosquito Habitat Mapper, we're trying to get a handle on that before it gets out of hand for us. So we're in a little bit of a good position compared to some places that have had these diseases now for millennia, because maybe we can monitor and eradicate much more effectively using 21st century tools. Okay. So anyway, another thing that I think is really interesting about this is we're collecting data about the Earth system. We're collecting data about changes in environment, but these are things that have immediate implications. If you actually identify mosquitoes that have the potential to cause disease, those disease mosquitoes are in our database, and then that data can be shared by public health officials, by mosquito surveillance officials, and by scientists who are doing modeling to try and understand how to best handle these invasive species. So all of that is really, really important. And so it's not, what's really cool about this is it's not just science for science sake, but it's science with an immediate human health impact. And that's what you'll be working on this summer. So here are some of our goals. What we've done is we've developed a tool that individuals and communities can use to reduce risk of disease. And you may not know this, but more than 50% of the US does not have a mosquito surveillance or control agency. And so in these places, it's up to the individuals to do their own mosquito control and their own surveillance. And so this gives people a tool. Another thing that I wanted to talk about is that besides creating these new data sets that can be used by modelers, by people like yourself that'll be conducting scientific research, we're also supporting NASA's efforts. They have a lot of satellites up in the air collecting all different kinds of environmental parameters, and our app allows them to use the science data to work on and solve some applied health problems that benefit humans. So these are really very, very, very important things. And last but not least, for those diseases for which there are no vaccines, and that would include chikungunya, dengue, yellow fever does have a vaccine, Zika, malaria, The only things that are available to us are finding out where the breeding sites are, taking care of those breeding sites, and educating people. And this app does all three, and that's why we were actually able, with the USAID, to test this app in communities at high risk for dengue and Zika in South America. And that's what I was doing the last 2 years. We can talk more about that later. Okay. So here's our team goal. What we want to do is we want to increase the spatial and temporal density of observations. Right now, there's about 12,000 observations on the site after our pilot. We really want to build that out so that we have a really robust data set that we can use for scientific analysis. So we want to have it in lots of places, and our SEES group is all over the US and also in different parts of the world, so that's very exciting. And one of the things that we want is to obtain frequent observations from the same place. And that's really the goal. Our goal as a research team this summer is to, if you, for instance, the first person that came on this morning was from Dallas. Well, if we can get observations from Dallas, and get those observations, say every week we have observations, that will tell us a lot more about mosquito habitats and breeding behaviors and the problems of mosquitoes in Dallas than if we have one in Dallas, two in Austin, 3 weeks later we have another one in Oklahoma City. So, but what we're trying to do is jumpstart the building of this database so we can do these things. So Parker, sorry to use you as an example in Dallas, but there are a lot of places where we need these measurements. And because it's a new program, every place we need these measurements. And so with this project, we're collecting data, but for a scientist like yourself, like myself, like Cassie, we're not really collecting data is really fun, but it's not why we do science. We do science so we can do something with the data. And so that's really the reason why we put the cohort together, is to gather together highly qualified individuals that are interested in doing some data exploration this summer with the mosquito data. And so you'll have a chance to learn to use some tools that have been developed by NASA, as well as some other sources that will help you to learn some analytical techniques, some visualization techniques, and you can also use these data that were collected as a group to look at a question of your own interest. Maybe you are interested in ecology and you'd like to know more about the ecology of mosquitoes and breeding sites. Or maybe you're interested in population dynamics of mosquitoes. Or maybe you're interested in the health aspects of mosquitoes. All of these things are things that you can conduct research on this summer. Okay, let's see here. So this is just a real quick show and tell about how to use Mosquito Habitat Mapper. And these are pictures that were taken last summer when I was actually in Brazil. But it's a very simple thing. For those of you who have already downloaded, you've seen this interface, but this is the interface of NASA's Globe Observer. What you do is you just click on Mosquito Habitat Mapper, and then it leads you through a series of questions. And by following those questions, you're collecting data using a protocol. And in science, it's really important that the data that you use is collected the same way by all the data collecting participants, because otherwise it's not comparable. So what you have in this app is a protocol that tells you how to collect, when to collect, how to report the data, and that way the data can be then collected in our big database in the sky and then shared with everyone. So to use the app, all you do is you go outside, you look for some places that you might find mosquito habitats, and basically any standing water that's going to be standing there for a week or more is a potential habitat site. And you would be surprised at the incredible the incredible numbers of habitats that we have found. It's really quite scary in some ways, but also intriguing that these mosquitoes are so very, they're so very well suited to adapting to the habitats that they have. So down here in the upper right, you see we found some in a drain and in a sidewalk. Over here on the left, we're in a bamboo forest, and where people cut bamboo for various uses. The places that's cut collect water, and that's a very favored place for mosquitoes, especially Aedes aegypti mosquitoes in Brazil. I don't know what it's going to be like, when you're in Austin or in Oklahoma City, but we'll find out. And then once you collect your sample, you can put it in a plastic bag, or you can take it in a cup, and then you sample that out. You put it on a paper plate, and then you're going to attach the microscope that you have received. And I don't know, I think some people have not yet received their microscope, but I know they're all in the mail if you haven't yet. So stay tuned for that. And then you can use the microscope, gives you that little bit of extra magnification, another 100 power, so that you can look at all the features on the larva. And using those features, you can say, this mosquito is not a problem. Thank heavens. This is one of those mosquitoes that doesn't potentially carry disease. And you can report that. Or once in a while, you're going to find, wow, this is a Culex. We know that Culex mosquitoes are one of the primary vectors for West Nile virus, which is a very serious problem in the US today. So you'll write down Culex. Or maybe you will find one of these invasive mosquitoes that are now such a problem for us, the Aedes aegypti, which is the yellow fever mosquito, or Aedes albopictus, which is the Asian tiger mosquito. And this app will allow you to distinguish those from other mosquitoes in that genera. Last but not least, bottom right, if you find a place that you found mosquitoes, or you find a habitat, we're asking you to actually dump out that habitat. Now, we're talking here specifically about the container habitats. So if you find it's obvious if you find a lake, or that has mosquitoes, or you have a wetland or an estuary, you're not going to dump those out and just say no. But if it is a container and it's possible to do so, dump out the water. If people are collecting the water for a specific purpose, like watering a garden, all we need to do is just make sure you put a net over it so that the mosquitoes can't get in or out. So you can cover the water, and that's another way to protect people from mosquitoes. Okay, and there's so many great questions. We're going to try and get to these questions in just a minute. So let's talk a little bit about the expectations, so you know what you're getting into here. And I know that this is a pretty rigorous opportunity to do science in conjunction with the NASA Go program. But we also know it's summertime and you're doing this, you're probably also working, you may have another internship, you may be doing other things. And so it's not a full-time thing. I think that's really great because then you can sort of combine it with your other interests. But we're hoping that you can spend about 2 hours a week finding and documenting places that mosquitoes can potentially breed. And I was looking at the questions and the answers over here in the chat. And when we say about 10 observations, what we mean is 10 individual observations of different places, because every week, it takes about a week for the mosquitoes to develop. So if you go to the same place every day, you probably won't get a lot of data, but if you check every week, you will be able to say something. So that's important. But when we say 10 observations in a week, we don't mean that you find 10 mosquitoes a week. We also want you to identify those habitats where there aren't any mosquitoes. And that's also very important because, if we just have, in our database, if we just have where we have mosquitoes and we don't have examples of where we don't have mosquitoes, it doesn't tell us very much. So every time that you say no mosquitoes in the app, it's a very important data point. And so it's a little bit more fun to be able to identify them, but you'll have plenty of chance to do that. So whenever you get a zero, please write it down and report it because that's really important. So what we're going to ask you to do at each site is to use the mosquito habitat mapper tool to identify the site, and if you have mosquitoes, to then identify them, and then quickly use the land cover tool to take pictures and describe the local ecology and environment. And when you see the land cover tool, and we'll have something online to help you through this as well, a tutorial, but it asks you basically just to take a picture up, take a picture down, north, south, east, and west. And that data is what's really important. They also, if you want to, you can also describe the data and say, okay, I'm looking out here, and it's about 50% trees. The tree cover is about 50% as I look up, and the grass is about 100% on the ground. You can make those observations, but that may be too time consuming because you have other things to do. But just taking the pictures is going to be very important for scientists. So please go ahead and do that. Okay, so there are what we have are 5 explorations that we are going to lead you through over the course of these next 8 or 10 weeks or so, and they're kind of like assignments, but the objective at the end of this cohort is for you to have a science a science research project that you can present in early September. And if you do these 5 explorations, you're taking steps that will get you through the process of having a pretty cool little project. So they're not just assignments for assignment's sake. They are helping to ground you in a project that you can do. So the first one is I was really excited to see that we haven't even assigned it yet and people are already doing it, is just to learn about mosquitoes where you live. And the instructions are in our base camp, and Cassie's going to show you that. But basically we're saying, okay, where are you? And how many mosquitoes are where you are? And of those, which ones potentially cause disease? And is there a disease problem where you are? So just learn a little bit localize your understanding about mosquitoes. And then what we did is we asked you also then to take that extra step that scientists do and to read a scientific paper that's somehow related to what you've done. And I saw that some people, I'm sorry, I can't remember your name right now, but somebody saw something from Doc Wasserman from the mosquito control in Colorado, and I want to let you know that he worked with me as I was developing this app and helped to make sure that the key that we have is right. So it was really great to see his name. But what we'd like you to do when finding those articles, there's so much out on the web, and it's really hard to find something which is a real science research article. And we want you to have experience looking at those, because that's going to give you things that you can actually then use in your own project. So I'd suggest if you have a library, you can use a search engine that your librarian can help you, a science search engine. But you can just use Google Scholar. And if you go into Google and you select Google Scholar, what it will do is it will provide you with lists of articles that were published in scientific journals. And that's kind of the sort of the high level reading that we're hoping that you will do. And not a lot of it, but just a few articles. And then that will give you some scientific peer-reviewed journals that you can use to cite in your project. So if you didn't do that for your from what you've already submitted, please go back and find a great science article that you'd like to read and summarize. And then we're asking you just to take a minute and write 3 or 4 paragraphs about what you read, so that other people don't have to read that article, but you can tell them what's there. And then if it interests them, then they can go back and look at it too. So it's sort of, you share it as a research team, we share what we know. So it's a lot less work for everyone. And that's what we do in my lab. And so I think it's a really good model. So anyway, back to these 5 explorations. The second one is we'll have you design a field experiment. And then the third one is we're going to have you read there's this mosquito stuff with citizen science is so new. There is not an article right now out there in the literature that really talks about using citizen science mosquito data and answering a question also using NASA data. But there's one. And then it wasn't citizen science data, it was actually people put out these traps. But it's a really great example of what we might be able to do with our data. So we'll look at that article together. And then finally, we'll look at some of the tools that we can use for data mapping and digital mapping, and how to do some data analysis. And then we'll share what we know with each other. And at the end of this summer, we'd like you to submit a research abstract and a poster about what you've come up with at the end of the fair at our SEES Virtual Science Symposium. And I can't follow these. I know that Cassie is, but someone said the reading assignment can be 1 or 2 paragraphs. Absolutely, it does not need to be long. But just think that you've read this article and you want to help someone else by not making them read it. So just give them everything that you think is really important. And if they like what they've read, they may go read it on their own. So that's what it is. So yeah, 1, 2, 3 paragraphs. That's really fine. You know, it doesn't have to be a book report. Great. Okay. So anyway, you have the option of taking what you've done this summer. You can just end there. But if you're still interested, or if it ties into other responsibilities you have at university, in preparing for university, if you want to do a science fair, this would also be a really great start for a science fair project that you might want to submit in the spring when those come available. So, so we have some structure here for you. We have some due dates to keep you on track. And we'd like you to, within a week or two, try to meet those. We know people are on vacation, so it's not like you don't get graded on this because it's a research team, but it's good if we're all pretty much on the same schedule. If you're traveling, and I know a lot of people are going to have like a vacation or two maybe during this time, it's okay. Just bring your mosquito stuff and make observations wherever you go. We're going to have some prizes for those who do 50 and 100 mosquito habitat observations. And then what you get from this at the end is that you'll get a certificate of participation that you can put on your resume for applying to colleges. Your research will be uploaded to the NASA Globe Observer website. How cool is that? And of course, you get to keep the microscope. So these are some of the things you get. And then if you decide to go on and do more, our team of subject matter experts are happy to continue to work with you even at the end of this project for those of you who would like to continue. So that also can be helpful if you want to do a science project. So anyway, there are some really good things that you can get out of this that will help you with your future career objectives and goals, and if you're interested in going on and doing a little bit of science or just getting into a good college. So I hope this will meet all your needs. So now, we only have another 15 minutes or so, but what I thought I would do is I would show you how to make a mosquito larva trap. And it's very easy, and you can make as many of these as you want. If you wanted to do 1, that's fine. If you want to do 5, that's fine. If you want to do an experiment using different kinds of bait to attract mosquitoes, for instance, or put them in different places, that already is a really interesting research design. But to build your larva trap, what you need is a drink bottle, like this one right here, a 1 or 2 liter pop bottle, and then you need scissors. You'll need a rubber band. You're going to need a piece of sandpaper, and I'm traveling, I don't have sandpaper, but I have a nail file, that'll work. And then you're gonna need to have, I'm trying to find it here, you'll have a little piece of nylon net. And you can buy nylon net like this. You can buy nylon net at a fabric store, but I've also seen that using something like a scrubby that you might have that has loops in it, or a bag that potatoes or onions come in, a couple layers of that is a no-cost way to obtain nylon netting. Okay, so to make this trap, This is very scientific and very technical. I don't know if I can actually accomplish this, but what you do is you take the bottle and you're just going to cut the top off. So you can choose anywhere you want to cut. Just cut off the top like this. Okay. And these are the two parts of your trap. In this part here, what you're going to do is you're going to put water. And the water is going to attract the mother mosquitoes. They're trying to find a nice, safe, secure place where water is still, where they can put their eggs. And so you're going to want to put this trap in a nice shady spot so it doesn't get too hot for the baby mosquitoes, the larva. And put some water in here, and then we'll talk about bait, the kind of bait you can use. You can put a little bit of fish food in there. You can put some grass. You can put, if you've got a pet bunny rabbit, you can put some alfalfa pellets in there, and we'll give you some more information about the kinds of things you can put in there. But anyway, you'll fill it up, fill the water up, basically to the top of where this mosquito, the top of the bottle goes in. And so here's the trap part. The mosquitoes can't get through this piece of nylon net. And this is basically, if you look at your window, this is window screen size. It can be a little bit bigger than that. Shouldn't be too much smaller than that. And all you do is you take that piece of netting and you put it over the mouth of the cup. And then you can take a rubber band, as I'm doing right here. Okay, you put a piece of rubber band on it to hold that piece of net in place. Okay, like that. And so what this is that the eggs can fall through into the water. But if you are gone for a week and the mosquitoes hatch and they turn into larvae, they turn into poop, and then they emerge as adults, the adults can't get out. So you're never going to cause an extra problem with this. This is a little safety feature. Okay? So then all you do is you make this a place that the mother mosquito is going to want to lay her eggs. And these container mosquitoes, there are culex that are container mosquitoes. That's basically the common house mosquito, the culex mosquitoes. Also the Asian tiger mosquito and the yellow fever mosquitoes. They're container mosquitoes. And so what you're going to want to do is you're going to want to give them a surface where the eggs will stick. And so you just take this piece of there we go, sandpaper, and just rough it up a little bit because that'll be a place for the eggs to stick. And then put the two pieces together like this, and it fits nicely. You don't need to tape it. And then you pour water in through this hole, and you have the water level to just below where the net is. And then what's going to happen is these mosquitoes are laying their eggs, and I'm talking specifically now about the Aedes mosquitoes. But these mosquitoes are going to lay their eggs on the side of the trap. Culex mosquitoes lay eggs on a raft in the water. So that would they would lay it just right down in there. Those are going to lay their eggs right on the side, and they're going to wait till it rains, and then their water rises up, and it gets the eggs wet, and then it falls through. And so you can have this set up somewhere in your you can put this outside on a deck or a balcony. You can put some in your house if you have mosquitoes in your house. You can put some outside in a yard, or even around garbage cans. If you live in a complex, you might want to put a little sign so people know what you're doing and with that. But anyway, that's your mosquito trap. To make it really attractive to the mom mosquitoes, you can cover it with like black paper or an old black sock, because if it's dark, it's even more attractive to them. But that's how you make your mosquito trap. All right, let's move on. Okay, so now we're through the the active portion of this project. Oh, okay. Let's see here. I'm just going to get out of this here. Okay. All right. So now we're ready for, we're here to answer your questions about the project. And Cassie's going to tell you a little bit about how things are organized so that you can see, so you can find how to connect and talk to other members of the cohort, how you can contact us and know that we're going to talk to you within 24 hours, Monday through Thursday, and also where to find resources. So, Cass, want to give them a short tour of the base camp?
Almost everybody that's on the webinar, of course, has accepted the invitation to join our base camp. And we have organized it according to the different themes that are here. So in our chat, it's kind of like an instant place to get an answer to a question. I really appreciate everybody who's introduced themselves so far and where they're from. It's such a great kind of a virtual community here. Within the chat, like I said, we'll reply pretty much instantaneously Monday through Friday. We'll probably reply to the chat. We have a threaded discussion, and a threaded discussion, these are the topics. And so if you, for example, have a question about an assignment, click on it, and we can see that 4 people or 4 responses have happened. It's a nice way to ask a question. You're not the only one who probably has that particular question. And so you can ask, and it'll get answered here by one of us throughout the week then. A couple of people have asked about links for different webinars. And in our calendar, the dates that have, of course, an indication by it, if you click on them, you'll see a little bit about what the webinar is about and a link to sign up or to go right to the particular webinar. If you want to save time, you can add that calendar, add the SEES calendar to your calendar, and it'll automatically populate and update it if something new is added or something has been changed. One of the more important areas will be the docs and files. And these are documents that we upload so that you have reference to them. The welcome letter and assignments are here, as well as joining the team. I just put a quick slide as to how you can join the team. And we are also working with the Globe Mission Mosquito. Once a month we put out a newsletter, and there's information here about the scientist blog that Rusty writes. There's tips and tricks. And then we also are scouring the news for articles about mosquitoes. So you can find the most current research that we've been finding here also. And let's see. The to-dos, we didn't necessarily start anything official, just to letting you know when particular milestones are kind of due, so you don't get all balled up at the end of the summer without anything to do. If you want to find or to talk to a particular person, the ping feature is the way you can do it, and it's just starting a private chat with somebody, and so that won't be documented within the base camp at all. Any questions about the SEES base camp so far? I'm going to stop this share, and I want to share another option of things and resources for mosquitoes if you'd like to see them. We have a website that's called Globe Mission Mosquito within the Globe program. And on it, or in it, we have Mosquitoes 101. There's a few docs and files and PowerPoints about mosquitoes and their physiology, their anatomy, what we're looking for. a list of webinars that we're also providing to an educator community, which you're very interested and invited to join, and then our citizen science webinars. You can get to past newsletters if you'd like to see it, how to get to the data that you're collecting and how you can look at it in our visualization studio. Additional resources we have some books and resources, we have activities, kind of how to do things. Meeting the team is people that are associated with the team. Science Cafe is where you'll get Rusty's blog. And I would, sometimes our globe site's not necessarily very responsive to, as you can see here. So kind of why I wasn't showing the whole thing. And hold on. Discussion, it's another place to ask questions. So if you're, depending on where you're at, if you have Basecamp loaded or you're on the web and you want to ask a question, we just provide you different pathways to get in to ask questions. If I want to show you how we find out what's going on with our teams, but the Globe team, I'm just going to show you what we've done, what you all have done so far. And I know we're about in the 5th one now. And you all can do this at any time too, but I just jumped to SEES 2019. We now have 55 people who have joined it. 5 people have taken cloud observations and 14 mosquitoes. That's awesome. And we can, as you zoom in around on the map, you can see where everybody is coming from. And if you want to see something specifically, you can look here. Mosquitoes falls under hydrosphere. You're a citizen scientist, so it's all anonymous. But if I click on a particular spot, it will take me out to the visualization studio and will populate with locations and photos if you've taken a photo, and information, only the Latin long, what kind of data you've taken and how many observations, so we know there's one observation here. Remember, no data is acceptable data, and that's possibly what we have here. Any other questions so far? Okay.
Okay, I think that was great. Thanks, Cassie. I know you probably still have a lot of questions because you have to still, some of you still have to download the app. Once you get the app, you have to explore it a little bit. What you'll see is that in both the Mosquito Habitat Mapper and in the Land Cover app, there's a little tutorial. And you're going to want to finish those tutorials before you can actually get into, well, okay, I have to step back. In the Land Cover app, there are 3 tutorials. You need to finish those tutorials before it will actually let you take data. In the Mosquito Habitat Mapper, it was developed differently so that the app itself is a tutorial. And so you can just start right away. And if you have a question, how do I sample? It will tell you how to sample. How do I look at it under a microscope? It will show you how to prepare your specimen and look at it under the microscope. So in some ways, the Mosquito Habitat Mapper is very much self-explanatory. But having said that, of course, you're going to have questions. And so there is a place in the Basecamp where you can pose a question about anything. I set up some threads, so it's a little bit organized, so you can find out where you can say what you want to ask and find that place and then put your question. We'll definitely try and answer them as soon as we can. But I don't think you'll really have any problems. Sometimes it's just getting to know know the technology a little bit. And I have to say, when I saw Cassie going in and looking at our team and then pulling down the globe visualization system and seeing those materials, it seems very complicated, but it's actually really quite simple, and you'll get to be able to do that very quickly in no time. And and being able to use these collaborative tools for doing collaborative science. I don't know if you've looked at many science papers, but it's not uncommon now to have a science paper with 21 authors. And scientists are very collaborative. We love working with other people. And I think that that's why this is kind of a very exciting project, because very often students don't always have a great, I know as an undergraduate, I didn't have a lot of experience doing collaborative projects. It wasn't until I was in grad school. I was like, oh, wow, this is really fun. So anyway, I think we're going to have some good times collaborating on these projects, using these collaborative tools, learning about technology. And here is a little message that tomorrow there is going to be a webinar, and we will make sure that you are aware of it, and we'll send you a note. But if you're available tomorrow at noon, at 12 o'clock Eastern Standard Time, Dr. Rebecca Boger College in New York is going to be about how to use mapping tools to analyze data. And all of the webinars that we have will be, you can access it at any time. So if you can't come, that's fine, but we'd like to invite you. If you can't see it though, it will be available and you can see the recording. So we'll have lots of opportunities like that. We'll let you know what's going on in our scientific community and let you know what's available and where you can find it. And I think you could make this as big as you want. It could be a 3-credit college level course if you took advantage of all the opportunities you'll have. But it's also summer and you may not want to work that hard, and that's fine too. Okay. If there are any other questions, please put them in the chat, and Cassie and I will stay online. But that's the official. And so much for taking time out of your day to join us and to learn a little bit about this project. If it's not your cup of tea, let us know too. If this is something that you think, it sounded cool in the paper, but now that I've seen it, I'm not sure I want to know. Just let us know if this is not right for you. And there's no harm right now in choosing to do something else this summer. So just want to make that clear. All right. Thank you so much. I'll be in minutes, but you're free now to go and have lunch. Thanks. Bye.